We finish with the lymphatic system by discussing secondary lymphatic organs and some of the immune cells in your body. And we should first think back to what primary lymphatic organs are, and those are where lymphocytes mature and develop. So the red bone marrow and thymus are where the B cells and T cells, respectively, mature and develop. And then those cells can migrate to other places in your body, like lymph nodes. Secondary lymphatic organs, like lymph nodes, are where you mount an immune defense. And so they are spread throughout your body in different places. But if we look at our list here, we'll see it includes the spleen, the lymph nodes, and then some structures that we call MALTs. MALTs are mucosa-associated lymphoid tissues. And the key term here is mucosa. We're saying that mucousy regions like your oral cavity and esophagus, your stomach and intestines, those mucousy areas have lymphatic tissue embedded in the walls. And so we'll look at some of the MALTs, specifically the tonsils, Peyer's patches, and the appendix. So spleen first, it is the largest lymphatic organ after all. It's found in your upper left abdominopelvic quadrant, and it has several functions. One is that it's a blood reservoir. It holds about a cup of blood, which doesn't seem like much, but I don't know that I've had an injury bad enough to bleed out that much blood. So in that event, the spleen would be able to release that much blood to help keep your blood pressure constant and to keep your cells oxygenated. The spleen also stores the broken down products from all of the red blood cells that it actually chews up, but it'll store away stuff like iron and platelets and things that can be reused later. And when you were a fetus, the red blood cells in your body were actually made by the spleen. Those are great functions, but let's talk specifically about, as a part of the immune system, what does the spleen contribute? And there's really two main regions of the spleen that we'll discuss: red pulp and white pulp. White pulp are these little spherical nodules, little balls of white pulp, and they sit embedded within the red pulp. So the spleen is mostly red pulp with isolated areas of white pulp. So when you think about red pulp, think about red blood cells, because it's in the red pulp that old red blood cells are chewed up and broken down and dealt with. Remember, we learned that 2 million red blood cells per second are dying and being replaced. So the spleen has help from the liver, but the spleen is really a big site of hemolysis, breaking down those red blood cells. And the way it does it is pretty interesting. The red blood cells have to squeeze through these windy sinusoidal passageways to get through the spleen. And if they aren't flexible enough anymore to make it through, they are destroyed by macrophages, these big eater cells that can engulf the red blood cells and tear them apart. In the white pulp is where we have white blood cells. We're not going to chew up old white blood cells here, but instead these white blood cells, they hang out around central arteries and watch the blood as it moves through. They monitor the blood for pathogens. So those are two major functions of the spleen, take place in the red pulp and white pulp. What about these lymph nodes? Well, they're these little oval-shaped capsules, less than an inch in size, and they are hanging out along your lymphatic vessels. So as lymph arrives at these nodes, it gets filtered, and then it exits the nodes. You have them throughout your body. You've got 500 or 600 lymph nodes, but many of them are clustered in certain places. You have a cervical cluster, an axillary cluster, and then pelvic and inguinal clusters. In all cases, they operate the same. Lymph arrives through afferent vessels, A for arriving, And then usually there'll just be one efferent, E for exit, vessel. So the lymph kind of backs up inside of these lymph nodes because of that. And as it's in there, it will move through both the cortex, the kind of outer part, and the medulla of the lymph node. And in the cortex, you'll have a bunch of lymphatic cells. And in the medulla, you'll have even more different types of lymphatic cells. And their goal is to monitor the lymph, to fight any infections that are found, whether it be a virus or bacterial infection, and to attack any cancer cells that may be found. Lymph nodes are pretty important, pretty handy. We mentioned before lymphadenopathy, the swelling of the lymph nodes in response to some infection or immune response. And lymphoma, you may have heard of, like Hodgkin's and non-Hodgkin's lymphoma. That's the cancer of the lymph nodes. Because cancer cells can ride around in the lymph nodes also are sometimes biopsied to check for signs of cancer. Like lymph nodes in the axillary region may be biopsied to look for signs of breast cancer. Because as the lymph leaves the breast, it would go into the lymph nodes. Here's a quick histological look at a lymph node. So you can see all these little spherical structures are found primarily in the outer cortex of the lymph node. Those are where these immune cells are proliferating. Okay, let's do our MALTs. In these mucousy regions of your body, you tend to have chunks of lymphatic tissue, including your tonsils. And you have 4 sets, although only 3 are commonly discussed. One set is the pharyngeal tonsils. Those are up in the upper region of your throat, in your pharynx, and they're sometimes called adenoids. You also have palatine tonsils, and those are the ones you can more easily see if you open up your mouth and say, ah, they're in the back of your oral cavity. You also have tonsils under your tongue, lingual tonsils. And so those are kind of the 3 most often known about or discussed or considered types of tonsils. There are also these tiny little tubal tonsils, and those sit next to the opening of the auditory tube, also known as the eustachian tube, which you may recall drains the mucus from your middle ear. So, if you were to have an ear infection or some bacteria that were starting to live in your middle ear, they might get drained away on that mucus, but now they're in your throat. What are you going to do with them? Well, there'll be these little tonsils next to that opening to kill that bacteria as it drains out of your middle ear. So those are your tubal tonsils. And tonsils are pretty cool. They're not these round little balls. They actually have like all these spiky indentations in them. And those are called tonsillar crypts. And the goal is to actually to trap some stuff in there. You want to trap maybe bits of saliva or food, and that will allow the tonsils to do their job, to do immune surveillance of the respiratory passageway. Since you have a lot of food, but also air moving through and past these tonsils, they can trap some of the stuff, see what's out there, and mount an immune response. Lots of things can go wrong with tonsils, as this image shows, partially stemming from the fact that these tonsillar crypts could trap some bacteria, and then an inflammation response could seal the bacteria in there. And now you have no oxygen and nothing to clean that area out, and you could get a bacterial infection or tonsil stones or other tonsillitis disease, and it's gross. Another MALT is called the Peyer's patch. These are about 1-inch follicles, so little spherical structures in the walls of the small intestine, especially the later small intestine called the ileum. And just like the tonsils are doing respiratory immune surveillance, the Peyer's patches are doing intestinal immune surveillance. trying to trap, monitor pathogens, and destroy anything they find. The other thing Peyer's patches do is make sure that some of the bacteria that live in your guts and that help you don't go and try to help you somewhere else. So, like, for example, we have E. coli in our GI tract. We need it. It helps us make vitamin K, for example. But that doesn't mean you want your E. coli taking a cruise and going to explore other parts of your body. So these Peyer's patches line the walls of the intestines, keep an eye on what's going past, and also try to make sure we keep those E. coli inside of us. And I have a few E. coli pictures in here. Just to remind us, here's some happy E. coli helping us. They're our friends. Symbiotic bacteria. Here's one being attacked by viruses inside a human host. You know, that's our pal. You don't want those viruses attacking your E. coli. So, although we tend to think about E. coli ending up on our salad bars, do remember they are essential for our survival. And our last malt is our appendix. This is a blind pouch on the end of our large intestine. We used to think that it was vestigial. It had no purpose anymore. But in fact, there is chunks of lymphatic tissue embedded in the walls of the appendix that helps with intestinal immune surveillance. Again, trying to trap and monitor pathogens and destroy anything that are found. So if you've had your appendix out, you may wonder, well, is my immune system now immunocompromised? And the answer is yeah, technically, like the same way that if you've had a tonsil or tonsils removed, you are missing a little bit of your immune system there as well. In general, there are some very, very specific strains of Clostridium that people without their appendix can get, but overall, it's a pretty minor contributor to your immune system. I probably wouldn't want to have like my appendix out and my tonsils and my spleen, although some people do. But just the appendix in general, there's not a massive immunocompromisation to the body. But one cool thing the appendix does is that as food and leftover waste move through your intestines, you could potentially one day have some type of intestinal infection. And the body's response to help you get over that infection would be to flush everything out of your intestines, meaning to have diarrhea, to carry away all those bad bacteria, to flush them out. But remember, you have good bacteria too, and you don't want to flush all that out as well. So the appendix serves as a little dead end where you can have some of your happy bacteria still surviving, and after everything else gets flushed out, you can repopulate your GI tract afterwards from the appendix. Probiotics or something could potentially also help with that, but this is like nature's probiotic. And of course, no discussion of the appendix is complete without mentioning the risk of appendicitis, where perhaps the opening to the appendix somehow gets blocked, gets obstructed, and you can get some fluid buildup in there. You can get ischemia, so a lack of delivery of oxygen, and now you'll have these anaerobic bacteria happy that there's no oxygen, and they'll start to grow, and parts of the tissue can start to die off through necrosis because there's no oxygen, and the appendix will swell, and you don't want that thing exploding inside of you because of the dead tissue, because of the bacteria taking over. So these emergency appendectomies are trying to remove the appendix before it bursts, and you have a lot of secondary infection risk. This is a really bad looking appendix, studied with the sonogram. I don't see anything wrong. It looks fine to me. But if you're into sonography, there are lots of websites that will train you on how to tell what a normal appendix versus a inflamed appendix look like. And that's just one example in that picture from the web. Okay, let's talk briefly about some of the lymphatic cells in your body. We've already mentioned these, or at least a few of these. T cells, remember, become immunocompetent, become mature in the thymus. So T for thymus. And they provide what's called cell-mediated immunity, meaning the cells go around and like fight other cells that they find. And they can fight a lot of different types of pathogens, and including cancerous cells. So they are your major portion of your immune system. Cell-mediated immunity. Other cells like B cells, remember, they mature in the bone marrow, and they make antibodies. So antibodies can go out, they can mark pathogens for destruction, and they can even like coat viruses and stuff to make them unable to infect your cells. NK cells are more rare. They're called natural killer cells, and they are conducting what's called immune surveillance. So they are monitoring your own cells for infection, maybe the infection of a virus, or maybe a mutation has caused the cells to become cancerous. So in either case, they're really focused on your own body's cells and destroying your cells if they get taken over. By the way, they're called natural killer cells, not because they attack your own cells, your very nature, but because they don't have to go to the I don't have a joke for that, right? There's no organ that starts with N where they go to mature. They are natural killers. They already know what to do. They don't have to go somewhere to be trained. So that's where their name comes from. So here's a slide that you can look at later if you want that just gives a little bit more detail on these three types of cells. But we should also mention that you can read a comic book for an even more soft introduction to those three types of cells. And here's just a peek at a comic called Viral Attack. It's very short, but you can see here an epithelial cell being infected by a virus and calling for help, and read about the immune response. One more special immune cell we should mention is macrophage. These are these large eater cells, and I like to say that they have a present for the T cells. And the joke here is we call macrophages APCs for antigen presenting cells. So they're going to present something to the T cells. Here's the story. Macrophages will wander around and eat stuff up. So let's say they eat up a bacterium. They will then stick that pathogen inside a phagosome, a little vesicle, and then they'll send over a little vesicle full of digestive enzymes called a lysosome, and they will use enzymes to degrade that pathogen. So they break it down into a bunch of tiny pieces, but then instead of just dumping out the waste product, they'll save some of those fragments, some of these antigens, and they will present them on their own cellular surface. Then T cells can come cruising over and check out what the macrophage has encountered, right? Because maybe this macrophage has been out wandering around and picked up a few different types of antigens. Now the T cell gets an alert. Hey, this stuff is out there. We should do something about it. And then, yeah, the macrophage will dump out the rest of the waste products through exocytosis. So that's just a tiny little glimpse into the immune cells of the immune system. If you ever have a chance to take an immunology course, I highly recommend it. Very interesting stuff going on. You will at least learn a bit more about the immune system in physiology. But for now, that's the end of the lymphatic system.